everyone, welcome to Swati's Bioscience Hub. So in the last two sessions, we have been discussing about the cellular respiration, okay? And in the last two videos, we have already discussed the first two steps of cellular respiration, that is glycolysis and pyruvate dehydrogenase complex, okay? Pyruvate dehydrogenase complex. The first step is glycolysis, where a 6-carbon compound glucose is a 6-carbon compound. One glucose molecule follows 10 enzymatic reactions or steps to produce 2 molecules of pyruvate, which is a 3-carbon compound. Okay, in glycolysis, glucose break down to produce pyruvate, and this pyruvate cannot directly enters into the Krebs cycle, okay, where it has to produce ATP. So its pyruvate cannot directly enters into the Krebs cycle. That's why in pyruvate dehydrogenase complex, this pyruvate converts to acetyl coenzyme. It converts to acetyl coenzyme, which can directly take entry to the Krebs cycle. Now, this glycolysis is independent of oxygen. It does not depend on the availability of oxygen. It can occur in both aerobic and anaerobic condition, and also it can take place in both eukaryotes and prokaryotic organism. And this pyruvate dehydrogenase complex, this pyruvate behaves differently depending on the availability of oxygen and requirement of our body. Okay, that we have already discussed in our last video of pyruvate dehydrogenase complex. So, in pyruvate dehydrogenase complex, there is a multi-enzymatic complex which converts the pyruvate to acetyl coenzyme. Pyruvate is a 3-carbon compound. In this process, it removes one carbon dioxide and it produces acetyl coenzyme, which is a 2-carbon compound. Okay, so these two steps we have already discussed, and today we are going to discuss the third step or the important step of cellular aerobic respiration, that is Krebs cycle. Okay, so today we are going to discuss the Krebs cycle. Krebs cycle is also known as citric acid cycle or TCA cycle. Krebs cycle is also known as citric acid cycle. First name is also Krebs cycle, which is also called as or known as citric acid cycle. Citric acid cycle or TCA cycle, tricarboxylic acid cycle. Okay. So first, why it is called Krebs cycle? Because Hans Adolf Krebs and William Arthur Johnson in 1937, these both people, Hans Krebs and William Arthur Johnson, they first identified the Krebs cycle or this process they first identified, for which they have received the Nobel Prize in 1953. They have received Nobel Prize in the field of physiology or medicines in 1953. That's why it is known as Krebs cycle, and also known as citric acid or TCA cycle. Why it is called citric acid cycle or TCA cycle, that we will understand when we are studying mechanism. So now we will discuss the mechanism or the step-wise pathways of Krebs cycle. Krebs cycle mostly takes part in the mitochondrial matrix of the cell, and it only takes place in the aerobic conditions. Okay, because only in aerobic condition pyruvate converts to acetyl coenzyme. That's why Krebs cycle only takes place in the aerobic condition and occurs in the mitochondrial matrix of the cell. Now this Krebs cycle, the precaution is acetyl coenzyme. Okay, acetyl coenzyme, which is a 2-carbon compound. Just a minute. So this acetyl coenzyme is a product of pyruvate dehydrogenase complex, which is a 2-carbon compound, and here it enters into the mitochondrial matrix along with some water molecule. Okay, and before it there is present oxaloacetic acid, or oxaloacetate. Okay, that is a 4-carbon compound. So now this oxaloacetate and acetyl coenzyme, they undergoes a reaction and produce citrate. Okay, citrate is the first stable product of Krebs cycle. That's why it is known as citric acid cycle. And the citrate consists of 3 tricarboxylic acid groups. That's why it is known as TCA or tricarboxylic acid cycle. Okay, it consists of 3 carboxylic acid group. 3 number of carboxylic acid groups are present in citrate, in the construction of citrate. That's why it is known as tricarboxylic acid cycle or citric acid cycle. Now the first reaction here is known as condensation. Okay, the first reaction is known as condensation. First reaction is condensation, and the enzyme responsible in this reaction is known as citrate synthase. Citrate synthase. Okay, in the first step, this acetyl coenzyme and the previously present oxaloacetic acid, they react together with water molecule and produces the first stable product of Krebs cycle, that is citrate. In the next step, this citrate removes water molecule from itself. Okay, it removes water molecule from itself and it produces cis-aconitate. Cis-aconitate, which is not a very stable product. Okay, which is not a very stable product. That's why the cis-aconitate immediately accept water molecule. It accept water molecule or get dehydrated, and here it produce one isomer of this citrate, that is isocitrate. Okay, and this is the isomerization process. The second step is the isomerization process. So here I am writing the second step is isomerization, and the enzyme responsible here is aconitase. The enzyme responsible or the catalyzes this isomerization process is aconitase. Now this isocitrate will undergo oxidative decarboxylation process. It will undergo oxidative decarboxylation process. Decarboxylation means it removes one carbon group, and oxidation, it will undergoes oxidation process. So here this isocitrate undergoes oxidative decarboxylation process, in which it removes one carbon group in the form of carbon dioxide. Okay, and when there is oxidation process is taking place, there must be something to be reduced. Okay, so here NAD reduced to NADH and H plus ion. So here it removes carbon dioxide molecule and it removes NADH. It reduces NADH, and after reducing, isocitrate converts to alpha-ketoglutarate. Alpha-ketoglutarate. In the third step, it is producing alpha-ketoglutarate. So these are the products at each step. So now this and the enzyme which is responsible for oxidative decarboxylation process. Oxidative decarboxylation process is known as isocitrate dehydrogenase. Isocitrate dehydrogenase. Okay. So now in the fourth step, this alpha-ketoglutarate will again undergoes oxidative decarboxylation. That means it will remove one carbon dioxide group. Okay, it will remove one carbon in the form of carbon dioxide and it will reduce the NAD to NADH. Okay. So here this alpha-ketoglutarate in the fourth step, it will remove carbon dioxide and reduce the NAD to NADH and H plus ion, and here it will produce succinyl coenzyme A. Succinyl coenzyme A. In the fourth step, the product is succinyl coenzyme A. Now, this succinyl coenzyme A has a thioester bond. Okay. In the next step or in the fifth step, okay, the enzyme which is responsible for the fourth step, that is oxidative decarboxylation again. The fourth step is the oxidative decarboxylation also, and here the enzyme which is responsible for this reaction is alpha-ketoglutarate dehydrogenase. Alpha-ketoglutarate dehydrogenase. Okay, it removes the carbon dioxide group and reduce the NAD to NADH. Now, this succinyl coenzyme A here, at this step, this is an important step because at this step, succinyl coenzyme A has a thioester bond which get cleaved along with the phosphorylation process. Okay. The thioester bond of succinyl coenzyme A will get cleaved along with the phosphorylation process. That means here it will remove one phosphate group and it will produce ATP or GTP. ATP or GTP from ADP or GDP. So here this is the only step. The fifth step is the only step where Krebs cycle directly producing ATP. Okay. Now this succinyl coenzyme A, after producing ATP or after the phosphorylation process, it will produce succinate. It will produce succinate. Okay. And the fifth step is a substrate level phosphorylation. Okay, the fifth step is a substrate level phosphorylation, and the enzyme responsible for this substrate level phosphorylation is succinate thiokinase. Succinate, because it cleaves the thioester bond of succinyl coenzyme A. So here it cleaves the succinyl coenzyme A thioester bond, and here along with the phosphorylation process, it produces ATP, and the final product is succinate here after the fifth step. Now, from succinate, this succinate here it removes one hydrogen group, two hydrogen molecules in the form of FADH2. Okay, succinate removes two hydrogen molecule in the form of FADH2 from FAD. Okay, that means it reduces the FAD to FADH2. And this number 6, this enzymatic reaction is known as dehydrogenase. Okay, dehydrogenation process, and the sixth step or the dehydrogenation part, dehydrogenation is catalyzed by succinate dehydrogenase. Succinate dehydrogenase. Okay, so here it removes one FADH2 group. Now, after removing this FADH2 group, it will produce fumarate. The final product after sixth reaction or sixth step is a fumarate. The succinate will convert to fumarate. Now, this fumarate will accept one water molecule. It gets hydrated. Okay, it accepts one water molecule, and this water molecule, it places the hydroxyl group to the carbonyl group of fumarate, and and it changes the bond of fumarate, and fumarate converts to malate, the arrangement of fumarate. So this water molecule places the hydroxyl ion to the carbonyl group and changes the arrangement of the fumarate and converts it to the malate. Okay, and this hydration, the sixth step is a hydration process. Okay, adding of water molecule. So this hydration process is catalyzed by the enzyme fumarase. The enzyme fumarase. Okay, so here this fumarate converts to malate, and lastly this malate will convert to oxaloacetate. Okay, this malate will convert to oxaloacetate in the 8th step, and here it also reduces one NAD to NADH. It reduces NAD to NADH plus H plus ion. Okay, and this is also a dehydrogenation process, and the enzyme responsible for the 8th step or the 8th dehydrogenation process here, dehydrogenation process, and the enzyme responsible here is malate dehydrogenase. Okay, malate dehydrogenase. Okay, after reducing the NAD to NADH, this malate converts to oxaloacetate, and again this oxaloacetate will accept the acetyl coenzyme A from the pyruvate dehydrogenase complex, and it will carry forward the whole process again. And here at only 5th step, where the succinyl coenzyme A is performing the phosphorylation process, at this stage only it produces directly ATP. Okay, that too per one acetyl coenzyme A, it produces 1 ATPs. Okay, so this is the whole reaction of Krebs cycle, the mechanism, the step-by-step reaction of Krebs cycle. First, the acetyl coenzyme A, it reacts with oxaloacetate with water molecule and produces citrate. This process is condensation, and the enzyme is citrate synthase. Now this citrate then undergoes dehydration. It removes water molecule, produces cis-aconitate, which is an unstable product. That is, this cis-aconitate again immediately absorb one water molecule and it converts to isocitrate. Okay, which is an isomer of citrate. And the second reaction, okay, the second reaction, which is an isomerization reaction, which is catalyzed by aconitase. Okay, enzyme aconitase. Now this isocitrate undergoes oxidative decarboxylation. It reduces NAD to NADH, removes carbon dioxide and produces alpha-ketoglutarate. Then again this alpha-ketoglutarate will undergo oxidative decarboxylation, removes carbon in the form of carbon dioxide, reduces NAD to NADH and produces succinyl coenzyme A. Then the succinyl coenzyme A will undergo phosphorylation process. The thioester bond of succinyl coenzyme will get clipped and it produce ATP. Okay, and also succinate. Now this succinate will remove 2 hydrogen molecules in the form of FADH2 and converts to fumarate. And the enzyme responsible here is for the conversion of succinate to fumarate is the succinate dehydrogenase. Now this fumarate will accept the water molecule and rearrange its bond where the water molecule places an hydroxyl group to the carbonyl group and it converts to malate. Okay, and this hydration is catalyzed by enzyme fumarase. And in the 8th step, the malate undergoes dehydrogenation process to again produce the oxaloacetate, and this oxaloacetate and it reduces NAD to NADH here, and the enzyme responsible is malate dehydrogenase. So this is the overall reaction. So if we look at the overall, the basic equation of the Krebs cycle, we can see one acetyl coenzyme A acetyl coenzyme में, wait, I'm removing the enzymes. So here we have discussed the Krebs cycle for one acetyl coenzyme. Okay, so with one, the overall reaction is one acetyl coenzyme plus water molecule, there is one water molecule, second water molecule, and here at the seventh step there is a third water molecule. That means 3 water molecule plus 1 NAD, 2 NAD and 3 NAD plus 3 NAD plus 1 FAD, okay, plus 1 FAD plus 1 ADP or GDP, okay, plus 1 ADP or GDP. These are the substrates, okay. 1 acetyl coenzyme, 3 NAD, 3 water molecule, 1 FAD, 1 ADP or GDP. Now it is producing 2 carbon dioxide molecule. One in the third step, oxidative decarboxylation. Another one is the fourth step, oxidative decarboxylation. So it is producing 2 carbon dioxide molecule, and from from water molecule it is producing only 1 water molecule. Okay, in the first half of the isomerization. So 1 water molecule. Okay, from NAD it is producing, from 3 NAD it is producing 3 NADH. 1, 2 and the third. Okay, so 3 NADH. From 1 FAD, in the sixth step it is producing FADH2. So 1 FADH2, and in the fifth step from 1 ADP or GDP it is producing 1 ATP or GTP. 1 ATP or GTP. So this is the overall or basic reaction of Krebs cycle. 1 acetyl coenzyme, 3 water molecule, 3 NAD plus 1 FAD plus 1 ADP or GDP is producing 2 carbon dioxide, 1 water molecule, 3 NADH, 1 FADH2 plus 1 ATP or GTP. Now, if you remember the first video of glycolysis, then in glycolysis, 1 glucose molecule produces 2 pyruvate. In glycolysis, initially we took 1 glucose, okay. This glucose in glycolysis process break down to produce 2 pyruvate. In PDH or pyruvate dehydrogenase complex, from 2 pyruvate, this pyruvate dehydrogenase complex produce 2 acetyl coenzyme. Because it produces 1 acetyl coenzyme per 1 pyruvate, okay, this PDH. So from 2 pyruvate we make 2 acetyl coenzyme. So when we are making 2 acetyl coenzyme from 1 glucose, that means from 1 glucose we will produce this products into 2. That means from 1 glucose, Krebs cycle produces 4 carbon dioxide molecule, 6 NADH, 2 FADH2, 2 ATP or GTP plus 2 water molecule. Okay. So from 1 glucose it is producing 4 carbon dioxide, 6 NADH, 2 FADH2, 2 ATP or GTP and 2 water molecule. So it directly producing 2 ATP or GTP and it is producing the NADH and FADH2. This NADH and FADH2 will enter into the electron transport chain part of the cellular respiration, where they will help in synthesizing the ATP. Okay, so this is the overall reaction of Krebs cycle. Today we have discussed the mechanism, the steps of the Krebs cycle and the enzyme which are responsible for the steps of the Krebs cycle and the final products of Krebs cycle. Okay, so if you have any doubt regarding today's discussion, please comment down in the comment section and like and share my video, and don't forget to subscribe my channel. Thank you so much.